So I have I've got the unfortunate task of being the last presenter before lunch, right? So I'll try to make this as entertaining as possible. Okay, so I'm a newish faculty member at IIT Delhi. I just came back after 10 years in the US, and now I'm looking to start what we're calling the molecular electrobiology research group here at IIT Delhi. So how do I click next on this? Yeah, so like I said, we're looking to start the molecular electrobiology research group at IIT Delhi, and you know, really it comes from this realization that interfacing electronics with biochemistry is now one of the grand challenges of the 21st century. You know, we have these tumor treating electric fields there, wherein if you apply the right frequency and the right intensity of electric field on cancerous tissue, this is an FDA approved therapy now, you can actually inhibit the cancer, ultimately forcing cancerous tissue to become normal. Wearable devices, electronic devices are already being used for interfacing, sensing, you know, blood sugar levels, drug concentrations. The Apple watches that some of you wear are an example of this bioelectronic interface. Another one, much older, is photobiomodulation, in which light, you know, good old light, is used to treat skin cancer. So photodynamic therapy is one of the oldest forms of cancer treatment, in which you inject a patient with laser light. You inject a patient with, you know, a pigment which gets photoactivated on being incident with laser light. So our research group really works at this interface with three aims. One is to study the electromechanical properties of proteins and their polymers. The second is to study the electrochemical properties of proteins and their polymers. And the third is to study how they interface with light. Okay, and so today I'll be talking about aim 3 primarily, which is how proteins interface with light. So this is these are just two cells. You can see here that there are three types of polymers. The ones in blue are actin filaments. We've had a couple of talks about that previously. The ones in red are microtubules, and the ones in gray are the ones we haven't actually seen any talks on as yet. Those are called vimentin filaments, intermediate filaments, because they're intermediate dimensions between actin filaments and microtubules. Somehow intermediate filaments always seem to get left out of the conversation, but they are very much members of the cytoskeleton. Okay, and these three polymers form the skeleton of the cell. Okay, on intermediate filaments, Dave White's. Okay, I will look that up. But yes, so the cytoskeleton plays structural roles inside the cell. Okay, so they provide the cell with shape and rigidity. They're crucial for cell division, where they literally exert mechanical forces against the cell membrane and against each other, forcing mechanically the cell to divide. A big favorite of mine is that microtubules and actin filaments form what's literally called a railroad for the macromolecular transport across the cell. So if you want to move something across the cell, such as the mitochondria from point A to point B, you do it along a microtubule. Okay, and this is why a typical structural biologist or biochemist studies the cytoskeleton. It's usually one of these three reasons. Okay, now the reasons I'm interested in the cytoskeleton, however, are completely different. You see, the cytoskeleton has very interesting electronic properties. Okay, like any other protein, tubulin and actin that make up microtubules and actin filaments contain an abundance of tryptophan, tyrosine, and phenylalanine, which absorb UV light and can re-emit this UV light. Okay, so that's the absorbance and emission spectra over there. There are short distances between these tryptophan, tyrosine, and phenylalanes, short enough to allow energy transfer and electron transfer to take place. Now there are other proteins where that takes place. The distances are short enough. But what really makes microtubules and actin filaments special from an electronic standpoint is the emergence of a lattice in a microtubule and an actin filament. And you see that allows long range order, and that allows these electron transfer events to become significant over long distances, right? And so there's recent work by Malvankar's group in Yale that shows that bacterial nanowires are actually highly conductive. Okay, so the question that I'm looking to ask is, are microtubules and actin filaments also, can they also perhaps act as hotspots for energy migration? Remember, they're also directionally polarized. What I'm trying to motivate here in a purely visual context is, if you have a photon that lands on one end of the microtubule, can you, through repeated energy transfer events, eventually transduce that energy into chemical energy at the other end of the microtubule or the actin filament? So can these repeated energy transfer events allow for microtubules, which by now we know act as transporters of macromolecules, can they also act as transporters of electronic energy? That's the question that I'm going to be asking over here. So there's two ways of transporting electronic energy, okay, by and large. One is the all famous FRET, and in FRET, the photon hits the tryptophan or tyrosine, and it excites the electron into its singlet excited state. Okay, if there's a tryptophan or tyrosine right next door, nearby, you have energy transfer that takes place where the singlet state hops to the nearby tryptophan or tyrosine. And ultimately resulting in fluorescence of its neighbor, which is why FRET is also often abbreviated to fluorescence resonance energy transfer. The other way you can transfer energy is Dexter energy transfer, in which case the triplet state is excited and triplet energy hops to the adjacent tryptophan or tyrosine, in which case, because it's a triplet state, you have phosphorescence. Okay, both FRET and DET are governed by what's called Fermi's golden rule, in which you have the rate of energy transfer being governed by the V value and the J value. So that's the coupling and the spectral overlap integral. So coupling is how close do the electron clouds get to each other, how much do they overlap, and the J value, the spectral overlap, is how much do the spectra of the neighbors overlap with each other. So the absorbance and emission spectra, that is, okay? So which energy transfer mechanism would even take place in the cytoskeleton, right? So what we did was we calculated the Forster and the Dexter coupling values. So we started out very humble, very simple. You have to be very careful in these calculations. And you can find the PDB files for tubulin, actin, and vimentin on the internet. And we downloaded these and we extracted the separation distances between all the aromatic residues. Okay. We then measured their coupling using the standard FRET coupling formula. Now you can see here that the V value is dependent on a variety of parameters. It's dependent on the R cube, that's the separation between the two aromatic residues. It's dependent on the refractive index, the kappa value, which is the angles between the donor and the acceptor, and of course that transition dipole moments. So a similar energy transfer through FRET actually takes place in the famous photosynthesis, right? Where chlorophyll, nature has optimized chlorophylls to have massive transition dipole moments, allowing for FRET efficiencies that are 99%. Of course, in tryptophan and tyrosine, FRET efficiencies tend to be 14 to 15% typically. So we're trying to examine whether that might be significant over here. So we calculated the FRET couplings, and then went ahead and calculated the DET couplings. Now, Dexter energy transfer, unlike Forster resonance energy transfer, it does not use transition dipole moments. Instead, it uses actual electron overlaps. So what's called the frontier electron overlap. Okay, so you have to have a physical overlap between the electron cloud of the donor and the acceptor, which is why the distance dependence is so important. So if the donor and the acceptor are so far away such that their electron clouds don't overlap, well, there will be no DET. There is no energy transfer in that case. And so we calculated these DET couplings through Gaussian, okay, and we quantified them and compared them to the FRET coupling values. So you can see here, so we explicitly calculated DET couplings for a variety of values, and you can see here, so the orange line and the orange dots correspond to the microtubules. The blue ones are the vimentin filaments, and the red ones are actin filaments. And what we found was that on a log-lin plot, you could fit it empirically to this equation and extract alpha and beta parameters, allowing you to essentially develop a molecular ruler for det. So you have a distance relationship, that's R, how the distance allows the V value, the coupling, to scale. Okay. On comparing the fret and det rate constants, we find that the det rate constants actually vary over a massive set of orders of magnitude in all three. So in tubulin, actin, and in vimentin, you can see that the det values are just all over the place. The fret values, however, are significantly higher than the det values, and they're also significantly narrower than the det values towards the higher side. So this clearly indicates that as far as the couplings are concerned, fret would be the way to go, right? The green is clearly higher than the purple. There's no brainers, right? Now, so we have the couplings, which is the V value. What about the J value? That's the spectral overlap integral, correct? You can see here that the opposite is true in the spectral overlap integral. In every kind of combination, but two, the spectral overlap integral of the triplet state is actually significantly better than that of the singlet state, which suggests that not fret, but the triplet state energy transfer, det, might actually be more significant in cytoskeletal polymers. And that could be the dominant energy transfer mode. So you can see that here visually. Okay, so when we interfaced the V value and the J value together to actually get the rate constants, we found that there was a set of Dexter energy transfer values that was higher than the fret values for tubulin, for actin, and for vimentin. You can use the energy transfer rate constants and balance them against the intrinsic phosphorescence and fluorescence lifetimes. Okay, and set up a kinetic Monte Carlo. There's somebody else over here, one of the earlier speakers who spoke about a kinetic Monte Carlo. So we went ahead and actually did that. So every time we would randomly photoexcite some random residue inside microtubules and have it either relax back to the ground state based on its lifetime or hop to its neighbor based on its energy transfer rate constant, right? And so if you have a high energy transfer rate constant, you tend to hop more with time. If you have a high as opposed to if you have a higher lifetime as opposed to the rate constant, you just fall back straight down into the ground state. Now, in a solar cell, in an organic solar cell, diffusion lengths of how far the hopping gets to typically tends to be greater than 50 nanometers. Okay, so they're very high. In a photosynthetic system, the chlorophyll example that I told you, it's about 500 nanometers. Okay, so nature has optimized chlorophyll-based photosynthetic systems to be extraordinarily efficient light harvesters. Remember the cartoon I showed you with the energy hopping along the microtubule? The question is, how far do microtubules get, right? And the answer, at least for fret, is about 8 angstroms in a tubulin dimer, and and a similar value for the microtubule. It appears that vimentin filaments, at least in the monomeric state, are the best. They're about 12 angstroms, but in the polymeric state, they fall back to about 9 angstroms. So yeah, unfortunately, the result is not 50 nanometers. We're looking into ways in which we might be able to optimize these values, maybe supramolecularly. If you look at Dexter energy transfer, remember the J value, the spectral overlap integral with Dexter energy transfer was higher, but the results are actually less impressive in the kinetic Monte Carlo. In vimentin, there is practically no energy transfer that takes place. In microtubules and tubulin, however, there is a significant energy transfer taking place. 6 angstroms, but 6 angstroms is nowhere near what you would need for a transduction device, right? So these are the values. It appears that among the singlet fret and the triplet det, singlet fret clearly is the way to go. values are generally low because the separations between the aromatic residues are actually too high for the Dexter energy transfer to be significant. This is in sharp contrast to the bacterial nanowires, where you have the tryptophans that are chalk next to each other, allowing for Dexter energy transfer to significantly take place. And now we're looking at the formation of, at the possibility of whether biological systems can form these J aggregates, which are these essentially super radiant states that form where instantaneous energy transfer can take place over long distances. And so in conclusion, among singlet versus triplet energy migration, it appears singlet state, FRET, does dominate over triplet state, DET. Cytoskeletal monomers and polymers have similar diffusion lengths. And among the three cytoskeletal polymers, tubulin and microtubules have the best photoexcitation diffusion lengths. With this, I'd like to thank you for your attention. We now have a nice, healthy lab in IIT Delhi with 4 PhD students, one of whom Prerna is sitting over there. I invite you all to look at her poster. And so far, it's been a very, very nice, pleasant experience. We've managed to get funding from the DBT in IIT. So let's see how things go.
Thank you. Questions? Yeah, Srishti.
Really great talk. Thank you so much. I had a question about how dynamics of actin might be affected, or any cytoskeletal component might be affected by energy transfer. So could it, for instance, trigger polymerization or trigger cytoskeletal remodeling?
Yeah, that's the question, isn't it? That's the question I was really trying to get at, is what would you trigger? So it could also be the unbinding of a kinase, not the unbinding of a map, right? Right now, unfortunately, because your diffusion lengths are so low, I doubt, just being brutally honest, I doubt anything is happening, okay? However, if we do get to a point where the diffusion lengths are significant, then what you're saying, I think, would become highly significant.
Like, for instance, as Adhya showed earlier, in T cells, actually, we see really fast.
So
in T cells.
In T cells.
Yeah, we actually see really fast polymerization. They're very different from any other sort of non-immune cells.
Mm-hmm.
So there we have seen that there are some sort of photo triggers.
Really? Are there any papers on this?
Mine. It's coming up.
Yours.
We're working on it, but actually we have seen that. And we
Fantastic. Is it out in the archive? Well, maybe we'll take this offline, okay?
So I was wondering whether that's possible, that if a system is fast enough, it could be because of energy transfer. We didn't think that the cytoskeleton can actually absorb light like chlorophylls or like.
It definitely can absorb it. The question is, can it transduce it to in a significant manner?
Yeah.
That's the, but I'm keen to talk to you later.
I've seen the numbers. It's less than nanometer, right? That
Yes.
energy. So it's shorter than the monomer of tubulin, for example.
Exactly. Unfortunately,
So,
yes.
Not sure. So also I have other question. If no one else has a question, for example, in DNA, also as a biological polymer,
Right.
people have measured similar
Absolutely.
things back in 2090s.
1989 was the first Jacqueline Barton paper. Yes.
So how is that compared to? I don't know the numbers.
Good question. Pros and cons, right? DNA tends to be floppier, which makes energy migration harder. Microtubules are extremely rigid. Somebody said the persistence length was in the millimeter, right?
Yeah.
Which means it's a lot easier because it's a straight line. You're not having to. The orientation between the tryptophans and tyrosines is, once it's fixed, it's locked in.
But
you don't have a tyrosine that's flopping around, and
there you have bases spaced 0.3 nanometers apart to continue.
Right. So the distances are lower, but okay. So if you want to look at this mathematically, what you're talking about, sir, is this, okay? This value is better.
Yeah.
You're right. Kappa is worse. So it's a trade-off. Yeah.
So what I remember from that measurements in DNA is that electrons hop.
Electrons hop, not electronic energy. So which is interesting, of course. Yeah. But there are problems with Jacqueline Barton's some of the findings there. Yeah, we can talk about it later.
Mother.
Yeah. So I guess one question which we can discuss later.
Sure.
But it was some some aspect of this difference between the Dexter and the Foster. This thing. But the more important question is that if these experiments are to be done in the cell, and one of the problems is in trying to figure out what the refractive index is in the cytoplasm.
Big problem.
Right.
Yeah.
And most people don't care about it. They use the same as the water, and that's a really bad thing because it comes with a very high.
So what we did was we looked at Kurutchev's 2010 paper, which had, remember, this is not only is this inside the cell, this is inside the protein. So it's even harder. And not only that, if a tryptophan is on the surface of a protein, it'll have a different refractive index versus if it's in the hydrophobic core. So it's a complete mess. And that is a problem. We went with one value simply because you need to go with something in a model. But I do understand that there are, at some point, you're going to have to. So we don't set it. Yeah, we're very smart about this. And this is something I'm very proud of. We actually explicitly calculated for every pair. So over here, we actually extract every single orientation angle. And so it's not the random of 2 by 3, which is what you'll find in a typical textbook. Yeah. So we actually explicitly calculate kappa in our calculations, just to be extra accurate.
Just a small comment. Microtubules also undergo post-translational modifications,
Yes.
which can change their compaction state. I don't know how much difference that will make, but for example, you can have acetylation of the microtubules. And also when microtubule-associated proteins bind, that also changes the microtubule compaction.
I have a counter question for you. This is a brilliant idea. Is there any PTM that adds on an aromatic to the, because that would help me.
Yeah.
Am I taking too much time? Sorry, is it?
Yeah.
Yeah, I mean, that's a great suggestion. Unfortunately, removing it would increase the distance even further. So, floppy, yeah. Yeah, it's also highly charged, right? Yeah, I wonder what's going on there. Yeah, I see your point. Yeah, but to come back to yours, sir. Yeah, I wonder if there's a PTM. Sorry, Minhaj Sirajuddin in STEM in Bangalore. Yes, I am. Okay. You are Dr. Minhaj? So?
No.
I'm sorry. Okay.
Okay.
Yeah. So we'll talk after the.
Any other questions? Otherwise, let's thank Aarat and also all the other speakers of this session. Thank you.